From Microbe TV, this is TWiEVO, episode number 77, recorded on April 25th, 2022. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey there, Vincent. Good morning or good afternoon, depending on where you're joining for TWiEVO today. I think we have a fun episode ahead. We have a live guest.
Yeah, joining us from the United Kingdom, Alex Cagan. Welcome to TWiEVO.
Thank you. Great to be here.
I think Alex is our first guest brave enough to do a live stream.
Yes,
Good for you.
Good to know.
Oh, and Nels is frozen up. He'll probably thaw out in a sec. It happens. So let's see. It is 11 a.m. here. Oh, there you are, Nels.
Oh, boy, yeah. So speaking of a live stream.
You got? I guess you didn't get your Ethernet plugged in, did you?
My Ethernet's plugged in, but it had a little hiccup.
Okay.
Just as we're lauding Alex for joining, no safety net live stream, my internet, of course, falls out. Hopefully I'm not too choppy here. Luckily, if I do, I guess, come in and out, there's two of you to keep the conversation going. This is the beauty of having a live stream guest.
We got backup. No problem. So it's, what is it, 5 p.m. over there, Alex? Yeah.
4 p.m.
4 p.m. All right.
Yeah.
And are you out of winter yet?
Well, you're never really out of winter in Cambridge, I would say, but it's pretty sunny today. Yeah.
Oh, so you're near Cambridge, is that right?
Yeah, that's right. Yeah.
Very nice, lovely place. Haven't been there in years. Hope to get back someday.
Yeah, you should come visit.
So we're here today to talk about a paper that Alex has recently published. Let me show you a screen cap just to give you a little hint before we get going. There it is, just out in Nature. Somatic mutation rates scale with lifespan across mammals. So we'll be diving into that with Alex. And as a virologist, Alex, I love mutation rates because we're always talking about them for viruses. I show a slide to my students of mutation rate versus genome size among DNA and RNA viruses, because there it's very different depending on DNA or RNA and genome size, right? It's cool. Today we're going to learn about human mutation rates. But first, go ahead, Nels. I'm sorry.
Well, no, not at all. In mutation rates among mammals. And, and as we've talked about on This Week in Evolution before, this is sort of like one of the core features of evolution is mutations, the fuel of evolution. And so you would think that we'd have a handle on the mutation rate, sort of a core parameter, and yet we don't. And so this is like, one of the really fun and exciting things here about Alex's work that we'll dive into.
Before we dive into Alex, let me just show you where people are from. I think this is so cool. So Rima is from Lebanon. Andrew is from New Zealand. Rune is from Norway. Isn't that cool?
Yeah. Welcome back.
Gary is from Colorado.
Gary.
Hey, look at this. Indonesia. Oh, my gosh. And then here's someone not far from you, Alex, Lester.
Awesome.
So I just love the world view of this. How many people all over the world are interested in science? Isn't that awesome, Alex?
That is fantastic. Hi to everyone who's joining live.
And we really appreciate it. And Frederick, Maryland, look at that. I know Frederick. Seattle, Washington. Hey, Lost Laboratory. Lost Laboratory is a fan of the show. Minnesota.
There we go.
I love doing this at the beginning because people also love to tell us where they're from, right?
That's right.
Okay, I'm sure there are more. Anyway, go ahead, Nels. Sorry to interrupt you.
No, not at all. So let's let's kind of dive right in here. I think we have a lot of ground to cover today. And in addition to being a rising star postdoc, Alex is also a great science illustrator, great science communicator. And so we'll circle back to that topic at the end of the show. But Alex, let's start with a little bit about you. Tell us about yourself.
Sure. Yeah. So I'm English. Let's see where to start. I guess university probably. My interests in this topic. So I took a kind of unusual route to getting into genomics, I would say. As a kid, I was always really interested in biology. But ironically, when I was at school, the genetics classes put me off a little bit. I was really into animal behavior. And ironically, the kind of more mathematical side of genetics didn't really appeal to me. I wanted to know, why does a animal hunt this way, that kind of side. And it wasn't until I was at university, I was doing anthropology at the University of Cambridge, so not far from where I am now. And I was more into thinking of doing social anthropology, so looking at how different human groups behave and, where that comes from different perspectives. But during that course, you have to take a module in biological anthropology, which I wouldn't have selected. It was kind of forced to give you a more well-rounded education. But then I kind of rediscovered my love of biology through that course. Had some fantastic supervisors, including Bill McGrew, who's a primatologist. And so that really turned my interest back to biology. And then I ended up specializing in biological anthropology. So after my undergrad, I thought I'd be a primatologist. So I went to a field site in Uganda run by Kevin Hunt to study chimpanzee behavior, looking at well digging activity just for a few months. And then I loved it. I love field work, and that's something I miss now as a desk-bound genomicist. But the field work was fantastic. But I during that time, I realized to get enough data from the field it took such a long time. And I'm amazed at anyone who has the dedication to do it. But I kind of was thinking about, well, there's this new rising field of genomics and all the questions you can answer there. And there's just a flood of data in genomics. So I ended up starting a PhD at the Max Planck Institute for Evolutionary Anthropology, with Svante Pääbo, who's best known for his work on Neanderthal ancient DNA. But I actually went to his lab because I knew he had this side project that I'd always been fascinated in, that I'd heard about during my undergrad, about these, people may have heard of these tame foxes from Siberia. So there's the farm fox experiment where they were interested in the causes of animal domestication. And so this old Soviet scientist, Dmitry Belyaev, had sort of tried to experimentally recreate the domestication process by taking farm foxes and selecting them for tame or kind of fearful behavior. And he'd also, people are less familiar with the fact he replicated that experiment in rats and in mink. So for my PhD, I was kind of looking at the genetics of those lines, particularly the rats, and trying to identify, could we see the genetic changes that caused the kind of changes in behavior. So that was kind of linking my initial interest in behavioral biology and then genetics as well.
Yeah, very cool. Did it work actually? Were you able to make some progress?
That's a good point. So it was a challenge, and that's sort of what led me towards my postdoc actually. So we found genetic differences between the lines, but there was just a real challenge of, so there's these fixed differences in the genome, but what do they mean? Which ones of them are due to genetic drift that have just drifted to fixation, and which are actually positive selection? And you can try different tests for selection, but you never really know, which are the true important variants. It did sort of lead towards this current research because we realized a good approach was a comparative approach. So look at many different species, domestic versus wild ancestor, and look for shared commonalities in the genome that have changed. So that kind of got me onto comparative genomics. But no, I would say that it was an exciting project, but you need so much functional follow-up on these variants to know which ones are actually, if any, are important.
Yeah, good point. That's tricky to put those together. I guess if you would have done, if the experiment was done in mice, then maybe some possibility of making some recombinant mice. Harder to do in rats, and certainly even harder in mink, and then harder again in foxes.
Exactly. So we kept an intercross going. So part of my PhD was breeding the rats. So then we got up to F10, I think, intercross. But again, I left before we had a chance to analyze that data. So those tissues in a freezer somewhere for a future project. But there would be a good chance there potentially to.
I'm curious
that
about
was the PhD.
how you selected for different behaviors. You would identify with a certain behavior and then continue to breed and select.
That's a great question. So a lot of I was involved in the behavioral selection during my PhD, and it involved putting a chainmail glove on, you would we had a special cage for the rats. You would put your hand into the cage. You would wait. You would be filmed, and then you would do a series of controlled movements: enter the cage, back of the cage, then try and stroke the rat, and then it would be scored based on, the very different factors. Like, did they vocalize? What did they show aggressive behavior? So it was a bit of a bias in the scoring, probably, but it was very clear when you put your hand in that cage, whether it was a tame or an aggressive rat, almost immediately. They had completely different reactions. The tame ones would kind of fall asleep under your hand, and the aggressive ones, you knew immediately you didn't want to have to spend the next minute with your hand in that cage, biting the glove, things like that. But
so the aggressive ones you would then take and further breed. And so how many generations did you have to do?
While I was there, we only went through about 10 generations. But I think that rat line, if I remember correctly, had been through about 70. I think it was about 70 generations of breeding. It bred a lot faster than the fox line. And I think their behavioral phenotypes were pretty starkly different between those two lines.
So, Nels, we have a question, which is good. Why is it more difficult with rats? You said to.
So not more. I think the experiments, and as Alex is pointing out, you could breed really quickly, get up to 10 generations, 70 generations, et cetera. But I guess I'm talking about the genetic tools. And so Mus musculus, the mouse, has just been a workhorse. Actually, thanks to some local efforts here, it turns out, at Utah. So one of my colleagues here in human genetics is Mario Capecchi, who is part of a Nobel Prize 14 years ago or something like that, for being able to generate recombinant mice. So you can knock out a gene, or now fancy ways of altering genes, expressing them in different tissues at different times. And so that gives you this genetic toolbox to go back. And as Alex is saying, like, if you have a candidate mutation or potential adaptation, can you just put that into a wild type background and recapitulate the behavior or the phenotype? And so a bit easier to do that with, I shouldn't say it's simple to do it with any mammal, but you have a whole sort of score of tools at your disposal in mice, which has made them a really powerful system for genetics. Is that fair, Alex?
Yeah, absolutely. And it's one of the interesting things in genomics, I guess. There's kind of two stages, at least for this project, of the research. In the first stage, where you just sequence the genome of some of the 2 different lines with different behaviors, you can find a whole bunch of candidate loci. But then the really hard work, I think, is the next stage, where you have to say, okay, let's follow up on some of these. And it could be a whole PhD project, just investigating one of these mutations, following it up functionally with that different toolbox. And at the end of the day, you don't know if that variant you've picked is a right one or not. So that's still a challenge, I think, is how you can do large scale phenotyping of multiple loci, to try and save people from going down kind of dead ends potentially.
Yeah, exactly. Totally agree. So you've already taken us, Alex, from your undergrad, like we were out in the field with the chimpanzees. Then you brought us into the laboratory, but with some slightly wild critters. Rats aren't so wild. We've domesticated these fellows, especially for the lab. But then now we're kind of going even farther. Like, now we're sort of pivoting. And so tell us about your transition from grad school to postdoc.
Yes. Absolutely. So after I loved the grad school project, but I said I was kind of thought, well, we did these scans and where to go from here. And at the same time, I would become aware, just, through the science, Twitter and things like that, about this new field of somatic evolution and somatic mutagenesis. And it seemed like I was very excited to hear about some groups, including the group I'm now at, that were applying these approaches from evolutionary genomics and genetics, which I love, to this field of somatic evolution and somatic mutation. And it felt just like this exciting new landscape that, for technical reasons, there hadn't been much data generated on somatic mutagenesis. So it's like, well, now the tools will suddenly become available, and there's this whole unexplored landscape of somatic evolution. And so it just seemed like a really good opportunity to take kind of the theory I developed from the PhD and start trying to apply those evolutionary biology concepts to somatic mutation. Which led me to the Sanger Institute, which has kind of pioneered a lot of the approaches for generating the data.
That's very cool. So we have a lot of listeners who are grad students, and actually, could we do a little behind the scenes here? So how did you, like, the mechanics of, as you were figuring out sort of the science and questions you're interested, how did you actually step, or like, how did you pick a lab, or how did that process unfold?
Yeah, sure. I have to cast my mind back. So the way it worked out for where I ended up, I saw there was kind of a landmark paper from Inigo Martin Craner's, my current supervisor. He produced this study looking at somatic mutation and clonal expansions in the skin, which is kind of this first study of human somatic mutation, where we saw that as you get older, these kind of mutant clones start to grow and colonize skin tissue. and I thought that was really cool. And I think he was just setting up his own lab, so he was transitioning from a kind of a postdoc to a junior faculty. And I saw, at the same time as this cool paper coming out, that his group was setting up and applying for postdoc positions. They were available. So I think I just wrote to him personally and said how fascinating I found the paper, and I had a couple of ideas of things I would love to investigate. I think even this project that we're now about to talk about, I wrote at the time saying it would be really cool to apply this across species, and they'd been thinking about it a lot longer than I had. They had similar ideas, so I got invited to interview through that. So it was a direct email to get in touch originally.
Yeah, and a good lesson there, I think, is when in doubt, reach out. Sometimes it can be intimidating to get in touch with PIs. Maybe it helps a little bit. Someone who's also just early in the career. I think that's sometimes the conventional wisdom is, I should go to a famous lab that's been established. This is a great example of how sort of joining a group at the beginning of it can really lead to a lot of rapid progress, a lot of good energy, all kinds of good things happening.
Yeah, I would just add that. So I think I've had sort of both experiences. So Inigo's team was very new when I started, but then for my PhD, Svante was already very well established. He was already director of the institute. That also came about from just writing directly, having known he was working on this project. And I was just very lucky. The PhD who'd been working on the RAP project was happening to wrap up at the time that I wrote. So I think there's always, making contacts, you can always just invite good fortune. You can't get lucky unless you write and see what's happening. So that worked out very well. And there's definitely advantages to being in an established lab versus a new lab. And they both have kind of great pros and cons.
Yeah, agreed. Okay, so you moved from Germany back to the UK and are now in the Martin Corina lab. And so we've already kind of let the cat out of the bag a little bit. We're talking about somatic mutations, but maybe we should define that, especially the word somatic, maybe that, and how that sort of differs from mutations that maybe we usually think about in the germline, mutations that are passed from generation to generation.
Yeah, absolutely. I mean, so that was definitely when I started in the lab. I had to get kind of used to a whole new vocabulary, because the way people talk about somatic mutations is kind of similar, but a bit different if you're used to normal kind of germline population genetics. And so, most people, when they think of genetics, are thinking about the variants that are passed from individuals. You get the germline mutations you inherit from your parents that are found in all the cells of your body. But somatic mutations are actually the mutations that you accumulate within the DNA of your cells throughout your lifetime. So actually, there's no kind of consensus like there is in germline mutation. It's not that all your cells are sharing the same somatic mutations. Each of your cells is kind of accumulating their own somatic mutations as you age. And there's all these cool dynamics that go on between stem cells, and then they divide, and you end up with little patches of kind of closely related cells. So you can sort of apply those principles of phylogenetics that we normally think about at the population level to populations of cells within your body.
So is it correct to say that every cell in your body has a slightly different DNA sequence?
So it's probably yes. The only reason so there are some cell types. What's fascinating we're learning is that different cell types have different mutation rates, and some of the cell types have very low mutation rates. So there's a possibility that some cells may be identical, but probably we think there's at least one or two mutations happening, at least between each cell division. So
sure.
I think it's fair to say that every cell in our body has a different genome in that sense.
Yeah, because the polymerase error rate is, I don't know, 1 in a million. So the size of the genome, every time it divides. Although many cells don't divide, like neurons don't divide, right?
Yeah, exactly. And we're still learning about some of those kind of post-mitotic cells. It's a bit harder to characterize the mutation rate. So there's still things we don't know. We haven't characterized the mutation rate in every cell type in the body. So I can't say with confidence that every genome is different, but I expect it probably is.
Okay.
Yeah, that sounds right to me. Yeah. So that brings us to the paper, which is entitled Somatic Mutation, or as Vincent brought up, Somatic Mutation Rate Scale with Lifespan Across Mammals. And actually, before we dive into the science, it's kind of fun, again, sort of behind the scenes here. So this isn't the first time that your work has seen the light of day. It was actually before peer review, before this really great splash in Nature just over the last week or two. You put it up on the bioRxiv, is that right? And maybe
that's right. Yeah, about a year ago.
Okay, yeah. And then maybe walk us through that process actually of peer review. How did that unfold?
Sure, yeah. So let me think back. So we put it up on bioRxiv, which was a fantastic thing for the paper. We got great feedback from bioRxiv, and it's just great to have it out there, not behind a paywall. So then we submitted it. The peer review process wasn't too bad, actually. We were generally positive, but with some, again, like as you'd hope, there were some great points that we worked on that helped us kind of refine the paper and be clear. What kind of one of the major changes between the preprint and the final version was we added a supplementary note on how these results might relate to kind of theories of aging, because we wanted to be really clear on that, to not kind of overstate the results. These are kind of, we're quantifying a relationship, but it's not necessarily a causal relationship, and just to think, but then to reflect a bit on how these results might impact how we think about aging. So that was kind of an extra section that we decided to add after we put it on the preprint server.
Yeah, that's cool. So, and I think this is like best case scenario, right? So the work gets out like a year early, so that people can see it, chew on it, give feedback, start that conversation. Then it still improves through the peer review process, and then has a nice sort of emergence here, which it sounds like you're getting a lot of interesting attention from your paper. Is that fair to say, Alex?
Yeah, that's fair to say. It's been a little bit kind of surprising, but fantastic, actually. So we're really happy to see how many people are downloading the paper. It's great.
Yeah, no, that's great. Kind of the conversation echoing back a year later here. And I think in part with some of the great science communication that you've been doing yourself. So we'll have to, I don't have them in the show notes yet, but we'll add some of these Twitter threads that Alex put together to both kind of explainers, but also really bringing in one of your incredible talents, which is science illustration, and then applying that to your own work. And so we'll get there in a minute. But for now, let's hit these somatic mutations. And you point out that, we've kind of been looking at this for a while in humans, but not so much in mammals. And so why is it important actually to start to make these comparisons? Why should we care? Why should we go after all of this diversity out there for a comparative study?
Yeah, that's a great question. So I would start, but even just by saying in human, what we know until recently and still now is a bit limited. So there's still a lot of work still to be done to even understand somatic mutation rates across humans. But there's only so much for some of the parameters we're interested in, like how somatic mutation rates might influence things like lifespan, aging. There's only so much diversity within our species that you can kind of look at to see that relationship. But once you start taking a comparative perspective, looking even across mammals, but then maybe in the future beyond mammals, there's just so much more diversity on some of those kind of life history traits. So there's just so much more you can try and understand about that connection between somatic mutation rate and some of these life history traits by taking a wider perspective than just looking at humans and looking at, if we think somatic mutation rate might be related to lifespan, why look at the 20 years, maybe variation you find in humans when we can be looking at hundreds of years across different species?
Yeah, that's great. And there is so much mammalian diversity too, as you're pointing out. I think so in lifespan, which becomes the theme of this paper, but also in things like body size, other things like that. So how did you pick actually the sample? I think it was it 16 or so mammals that you decided to analyze here?
Yeah, that's a great question. So I think kind of the main challenge at the start of this project was getting access to tissues that we could actually study. And there were a few kind of restrictions on what we could study that kind of shaped the project. So first of all, we needed a tissue type. We knew from human studies that were emerging that the mutation rate varies across different tissue types in the body, different cell types. So we wanted one cell type, or tissue type, that we could compare across mammals. We decided on colonic crypts. So these are kind of crypts in the intestine for a few reasons, primarily because they're easy to identify histologically. So when you make a histological slide under the microscope, you can see these crypts. They're like little U shapes. We knew from humans that they were clonal units. So you have stem cells at the bottom, and eventually one stem cell dominates the crypt. And as it divides, kind of its daughter cells populate the crypt. So if you laser microdissect, which is a really fun lab technique, just with a kind of laser microscope, you cut out individual clonic crypts and you extract the DNA and sequence them. All of those cells will share the mutations that had occurred recently in that kind of the history of that stem cell at the base of the crypt. And so that enables us to kind of count those somatic mutations. Ideally, we'd have just taken single cells, but the error rate is still too high in single cell sequencing to really differentiate for sure between somatic mutations and artifacts. So we're kind of limited to clonic crypts. And so we thought, okay, well, where can we get clonic crypts from different mammalian species? And and not just different mammalian species, but for each animal, we need to know the age of the animal when it died to calculate the mutation rate. So if you say, okay, this giraffe cell has 1,000 mutations, you need to know how old that giraffe was when it died to figure out that mutation rate per year. So zoos turned out to work very well for that because they have great records on obviously the age of the animals they have. And also when the animal dies, you can quickly do a necropsy to kind of just, they do that to ascertain the cause of death, understand the health of the animal better, but then they can also collect tissue samples for research. Whereas in the wild, your chance of coming across an animal where you know its age and it's died within 24 hours before the intestines kind of just decomposed is almost impossible. So we were kind of limited that way. We set up some collaborations with different groups, London Zoo, and a few kind of for other species, particular research labs that were working with different mammals. And then we kind of had to wait opportunistically for when, the animals died of natural causes. We would then have these teams ready of pathologists at these different institutes who knew what we were doing and collect tissue samples for us. And we ended up with these 16 species.
Now I understand why you don't have any whales in there.
Yeah.
Yeah. Well, I would love some whales for the future.
Yeah.
So there are whale samples. One of our collaborators is this cetacean stranding investigation program. They describe themselves as like CSI for whales. So whenever a whale or dolphin beaches up, they'll go and do a kind of detective report to figure out cause of death. But the challenge is you see a giant whale beach, but how old is that whale?
Sure.
So that's still a challenge, unfortunately.
I suppose you could get some elephant samples from zoos, right?
Yeah, that's where I'm working on that right now.
Good.
That's the next phase. Yeah.
Very cool. And actually that gets to one of the potential themes here. So there's this sort of famous paradox in science, Peto's paradox, speaking of elephants. And so it's this notion that, animals with a lot of cells that are big, there's the chance for more somatic mutations, just sort of a numbers game here. And so Alex, maybe walk us through that. Peto's paradox, what is it? And why was that on your radar for the study? Yeah.
Absolutely. It was one of the main hypotheses driving the study, actually. So that this observation from Peto, kind of biologist or mathematician, that when you look across species with different body sizes, you would expect, like as you said, whales and elephants should be getting cancer much earlier. In fact, I think whales have quadrillions more cells than we do, these large bodied whales. They shouldn't even be able to reach adulthood without getting some kind of cancer, and yet they do. So there's this kind of paradox that cancer rates seem to be constant across species, despite differences in body size or lifespan. And so there's different potential explanations for that paradox, kind of evolutionary explanations. So you could imagine that evolution has acted to increase cancer defenses or kind of cancer suppression in these larger lived species. But what mechanism is that? And one potential mechanism through which they may have done that is to really reduce the somatic mutation rate in those larger species, because if mutations aren't happening as quickly, there's just less chance of cancer happening from accumulation of these deleterious mutations. So we wanted, when we were collecting these samples, to have a range of mammals with both different lifespans and different body sizes, so we could address the kind of the body size for the Peto's paradox question and lifespan for the kind of general aging question. And we also really wanted to try and find mammals that aren't necessarily strongly correlated on body size and lifespans, which is a challenge. Most large animals tend to live a long time, and most small animals generally tend to have shorter lifespans. So we were very happy to get the naked mole rat in this project, which is like a superstar species for cancer and aging biology. And it has a really long lifespan, I think 20 to 30 years, but it's about the size of a large mouse or a small rat. So where we tried to get species like that would enable us to kind of tease apart those two different variables of body size and lifespan.
Wow, that's amazing. I mean, a mouse is 2 to 3 years. I don't even know in the wild it's probably shorter because they get eaten, right? But 20 to 30 years, that's amazing.
That's really cool.
And until recently, people thought they couldn't even get cancer. Cancer's so rare. They've now seen it a couple of times, but it's so rare they're almost impervious to cancer.
Nels, didn't we have someone on who said that elephants have amplified P53?
So this was Vinnie Lynch.
That's right.
Yep. Who's now, I'm trying to remember if he's up at Syracuse. Buffalo, I think. He was at the show when we spoke to him. I apologize. I think my internet is still kind of hiccuping here once in a while. I'm on a direct Ethernet line, but it's been a little choppy. So if I sound confused, or I should say more confused than usual, that might explain it. I'm blinking in and out here. But yeah, so elephants so that's one of the hypotheses, right? For why, and actually there probably still stands. So having all these extra copies of the P53 gene, tumor suppressor gene, could that speak to how you get away with these extra trillions of cells over life? And because I think one of the punchlines from Alex's study is that actually it doesn't look like mass, that there is a big reduction in somatic mutation rate, that it's actually kind of a level playing ground, which sort of leaves open, like, is actually a really important result to think about, okay, what else could be going on here? And that, I think, is one of the current hypotheses, is that you start to evolve a number of sort of anti-tumor countermeasures, and these extra copies of P53 might be an example. So Alex, you mentioned this really cool technique that you have, laser micro dissections. So I guess you've got the tissues are coming in. So the animal at the zoo or related collection has died. You have the team of pathologists sort of at the ready getting this together. And then are you like literally under the microscope yourself, then doing a laser dissection and pulling this up? Maybe walk us through a little bit of the analysis.
Yeah, I'll walk you through the process, because it was pretty exciting to be part of that as it was developing. So it was originally developed for human, and then luckily, mammals are, at the cellular level, it's basically the same. So you could apply that same human pipeline to all the mammal species. So yeah, our kind of collaborators would get a little bit of tissue for us, like a kind of 1 centimeter cube of the intestine. They would fix that for us in a commercial fixative called Paxgene, which is kind of a ethanol-based fixative. Send it to the Sanger. Once it gets there, we would go through a kind of histology processing. So you embed it in a kind of paraffin wax. And then our histologist would kind of, with this, like, it looks like a tiny, like, salami meat slicer microtome, just slice very thin sections for us, which then go on a special kind of slide. So not a normal glass slide, but a glass slide with a plastic membrane, which is kind of compatible with laser capture microdissection. We stain it with hemotoxin and eosin, so that's what gives it that kind of nice pink-purple color, so we can see it under the microscope. And then, I was operating the laser microscope, and it's very fun because, as you mentioned, like, I like those to kind of illustration as you get in the lab. So we actually had a whack-on tablet where you, with a pen, draw around the crypts that you want to cut out, and then the laser just follows it. It's a Leica microscope, and the laser beam just cuts it out. Yeah, and again, when I just joined the lab, this was quite low throughput, so you'd kind of do it in individual tubes, cap each one. In fact, it was harder than that. They would fall into the caps, then you'd have to flip the caps over to put them on the tubes, and you'd lose about half of your little hard to see by eye cells, because they would probably just fly away with static when you tried to flip them. But we actually got a custom built stage in that could hold 96 well plates, which enabled us to do kind of high throughput processing of these.
So when you cut, you're also cutting the membrane, so the section just falls?
Exactly, but half the time the section doesn't fall. They're so small that they'll just stay by static, and you have to use a process called laser catapulting. So you increase the aperture of the laser, and then you just blast it with laser beam, and you hope that just fires it off into the tube. And you hope it's in there. You squint at the tubes, and you like sometimes see a dot, but you kind of just submit enough samples, and you hope that there's some cells in there when you digest them.
Years ago, Nels, we had someone from Charlie Rice's lab who was doing this for hepatitis-infected liver, right? And you could laser capture individual cells and look at whatever you wanted to in each one, right? You could look at viral genomes, cytokine responses. So cool.
Yeah, I love this use of tech, like putting this technology to use into, like, for really different questions. Shows how powerful it is. That's so cool. Yep. Okay, so you got hopefully enough of these get into your tube or your 96 well plate. They're not all sticking to the membrane. Then what happens?
So then we just digest the cells, lyse the cells to get the DNA out, and each tube will have a different individual clonic crypt in it, hopefully. And then we send it off. Sanger has an amazing sequencing capacity, so we have a pipeline that will then do library preparation for Illumina sequencing, and then about a month later, we get back the sequencing data. We map the reads. So that was another limitation I forgot to mention, is that for these species, we wanted them to have a reference genome available, because we need something to map the genome to. And that was quite limiting when I started. So Sanger has the Darwin Tree of Life program, which is part of this global effort to make a reference genome for every species on the planet, which is just opening the door for kind of what species you can study genomically, which is fantastic for going forward. But at the time, we got a lot of amazing samples from the zoo that we just are still in the freezer, because there's not a reference genome yet for us to analyze it. But so we would get the data back, map it to a reference genome, And I should mention, so somatic variant calling is a little bit different to germline variant calling. So one of the challenges you have is you actually have to filter out the germline variants, which is what you're normally looking for when you do genome sequencing. So as well as sequencing clonic crypts, we would also kind of take another just like bit of tissue, like the smooth muscle that you'd actually see on the slide around the intestine, and sequence that. And then basically during the variant calling process, you can identify those genomic variants that are found in every single crypt and in your kind of match normal control sample of like smooth muscle. And those would be the germline variants that that animal has in all its cells. And so we would filter those out. And then if we had multiple crypts from an individual, we could then just say, well, for each crypt, what are the unique mutations in that crypt that aren't found in other crypts or in another tissue type? And those would be our somatic mutations. And we could basically just count those, if that makes sense.
Yeah, it does. And then you also, as you pointed out, you have that time series, right? So you've obviously not from the same individual, but of the same species, maybe even, I'm guessing maybe some like same family members in some cases, if it was at a zoo or something like that. And so you have then you're piecing together, I guess, you're waiting to get enough samples that will fill out a time series for each species. Is that right?
Exactly. So not all species, we didn't have the luxury of a time series for some animals, like where they're just, they don't die that often and there aren't that many in zoos. But where we could, for mouse, dog, naked mole rat, and a couple of other species, we really wanted to get that time series, because when we began, we're like, well, no one had really called somatic mutations to different mammals, and we're like, well, can we really do this? How do we know what we're seeing isn't just artifacts? And there were a few things that really guided us to give us confidence in the results. And one of the key ones was having time series. So, a pattern you'd expect from real mutations, but not from artifacts, is that you would have a kind of a linear increase in mutation rate over time. So it's really reassuring when we saw for those species with like a 1-year-old mouse, 2-year-old mouse, that you see kind of twice as many mutations in the 2-year-old, because if there were artifacts, you wouldn't expect that age correlation. And another thing that was reassuring, it's again why we chose mammals, is that we already had an expectation of the types of mutations you see in clonic crypts from human work. So we could kind of see, well, are the patterns of mutations in a trinucleotide context, are they similar with what we see in human? Which didn't necessarily have to be the case, but it kind of gave us an expectation that could help us guide understanding if the variants we saw are likely to be real or if they were artifacts, which is kind of always the main fear, I think, in the genomic studies that I've done, is, just knowing that what you've sequenced, are these real or are they artifacts, before you try and make any conclusions about the underlying biology.
Yeah, no, and that's where having these time series, like, so in figure 1, you just see the rise of the mutations as it correlates with age. I mean, and then hitting these different sort of patterns orthogonally, really powerful. So you mentioned, some of these signatures you're looking at human mutation data. So there are these, I guess they're called cosmic profiles. These are, maybe explain for us a little bit about this.
Sure, yeah. So it's really fascinating. It's something that I had not come across before I started working on somatic evolution, but there's it comes originally from sequencing different cancer types and tumors, and looking at the the cancers tend to have really high mutational burdens because they have all sorts of things going wrong in the DNA repair machinery. And they notice from looking at different tumor types that you'll get very different patterns of mutation in different tumors. And so by patterns, I mean from a trinucleotide context. So every time there's a single point mutation, you can look at the base pair before and after it, like is it a C and a T afterwards, and that gives you the trinucleotide context for a mutation. And they notice in different tumors, you'll have very different kind of frequencies of kind of deposition of different mutations along the genome. Like some tumors will have very high amounts of C to T mutations at certain contexts, and others very different. And so they developed the process where you can kind of take your genome with all the mutations, and you can look at, well, what are those different trinucleotide contexts the mutations fall in? And then you can kind of compare it to a database, or you can do de novo extraction, but you can compare it to kind of known signatures that have been observed in different cancers and try and say, okay, the mutations we see in this genome match these processes, these kind of mutational patterns that we've seen before. And so kind of just to give some examples of that, this kind of smoking tends to have a particular trinucleotide context. UV damage is very detectable. And then there are kind of processes that we see just happening across the human body that seem to be kind of ubiquitous mutational processes that will happen in any cell type, even healthy tissue. And these are the kind of 3 of these processes that we see in the clonic crypts. So one of them is spontaneous deamination of 5-methylcytosine, these C to T mutations at CpGs. And so those seem to be kind of the dominant mutational process that's happening in clonic crypts, and they happen at different levels at different cell types in the body. Another one is oxidative damage, which tends to be C to A mutations at certain contexts. And then the other one we call signature 5, And this one is kind of a very flat signature, so it doesn't really particularly stand out in certain different genomic trinucleotide contexts. And it's also very similar to a profile we see in the germline. So we think what this represents is kind of an amalgamation of different processes of damage and repair, but we can't really separate them because all these processes are happening across all cell types. So the only way you can kind of really pick out what the different processes are is if you would have cell models where you've knocked out certain kind of ubiquitous processes, and then you'd notice, okay, this part of the mutational process is due to this repair process. And there are groups working on that, but we still have a lot to learn about just that kind of milieu of different constant ongoing processes of damage and repair.
Yeah, it seems like a really useful technique or way of analyzing the data, right? Because you have just scores of mutations, and how do you sort of squeeze some meaning from it? Vincent, remember we considered this a little bit previously on TWiEVO. We were talking about contagious cancers in dogs, this dog venereal tumor, and sequencing samples from around the world, and using exactly with some of those profiles that came from tumor samples originally, and that UV, it was interesting, the UV damage correlated for the dogs that were living closer to the equator. There was like a higher amount of that. And then the ones that were like farther away, there was less of the pattern. And so you see this like biological signal there. It's really interesting. Yeah.
I have a funny little aside actually relating to that. So one of the authors on this paper is Liz Murchison, whose lab focuses on the transmissible cancers in dog and Tasmanian devils. And I actually went to do some field work for her a couple of years ago in Chernobyl, actually, because they're trying to collect samples of this transmissible cancer that's been passed, from dog to dog around the world. And what's really unique about that dog transmissible cancer is that it has a very low mutation rate. So most cancers just accumulate thousands of mutations. But the genome of this transmissible cancer in dogs is very low burden, seems very stable. So, but it's found in kind of wild dog populations around the world. And so Liz had this idea that perhaps in Chernobyl, where there's higher background radiation, it'd be interesting to see how that cancer behaved if it was exposed to a mutagen. So I went there with a group that was kind of working to look at the health of the dogs in the Chernobyl exclusion zone to kind of vaccinate them. And while they were being checked, we looked for transmissible cancer, but actually it's one of the few groups of wild dogs where we didn't see there was no transmissible cancer there. So it's probably just random, but there was the idea that maybe those cancers are very responsible to radiation therapy. So there's always the kind of wacky hypothesis that the radiation might actually be protecting them. But yeah, it's just an aside.
Yeah, that's wild. Yep. So, and you mentioned one of the signatures, I guess, that you see and specifically from the crypts, from the intestinal cells, if you collect them from humans, is the impact of another passenger there, the E. coli. So there's a genotoxic factor from E. coli that's gives a specific signature. But did you see that in your mammals?
Yes, that was really interesting, actually. So we know that occurs at a certain low frequency in human crypts. This E. coli produces colibactin, which is genotoxic. And so we thought, we were also interested, would we see other new signatures in other mammals with different diets? But we didn't. And we actually had enough mammalian samples to calculate that there's actually a significant lack of this E. coli signature. And we don't really know why that is, but there's kind of some ideas that that this kind of E. coli that is genotoxic is just more common in the human gut. And that could be related to diet. I think there's an interesting study that we cited that actually if you give a cooked diet to mice, they have a slightly higher prevalence of this type of E. coli in the gut. So there could be a kind of role there of diet in what kind of E. coli colonize the gut and which strains are genotoxic. But yeah, it's kind of an interesting observation. We don't really know what to make of it. But yeah, it seems that other mammals have less of this genotoxic E. coli than we do.
Yeah, no, and your point's interesting too, just thinking about the different diets. I have a colleague here at Utah, Denise Deering, who works with these African rats that basically eat creosote or something something like totally poisonous. But but I guess, would you predict, so the fact that they can do that, I guess, maybe they figured out how to detoxify this before the the damage could happen, or that you've somehow protected the genomes of the somatic cells. What do you think about that?
Yeah, it's fascinating. So I guess a caveat with our study is these are mostly zoo or lab they're all zoo or lab animals pretty much. So they're not really probably potentially exposed to some of the kind of really strange diets that are out there in the animal kingdom. But it does seem that these colonic cells are pretty good at protecting themselves from. We're only mostly just seeing endogenous signatures, apart from a few exceptions. So there could be, I guess, it's the role of our body cells in protecting themselves from genotoxic damage. But I also wouldn't be surprised if, as we look at more species, particularly from the wild, that we may see some signatures. But I guess you particularly think the bladder is also somewhere where you might see toxins. It would be interesting to look there as well.
Yeah, I agree. Really cool. Okay, so we should talk about actually the flavors of mutations here. So you're thinking about the point mutations in the context of the trinucleotide, but of course mutations come in all shapes and sizes, like literally different sizes, copy number, things like that. So maybe give us a sense of some of the other stuff going on there.
Yeah. So I would say we focused on the single nucleotide mutations because they're the easiest to actually confidently call in the genome when you're doing variant calling. We're better able to differentiate between artifacts and real mutations that way. But we did want to look at structural variation, kind of these larger scale changes. But there's many more challenges in that. So the quality of the reference genomes we were looking at are very different across the different species. And so when you have reference assemblies that, instead of kind of assembling whole chromosomes, are just very small contigs or scaffolds, it's a lot more challenging to confidently call. If there's a big structural variant that's larger than your actual contigs of the genome, it gets very tricky. So we were less confident about structural variant calling, but from the species where we were confident in the quality of the reference assembly, we saw a very low number of structural variants, which was in line with kind of what was known from humans so far as well, which is that structurally, clonic crypts seem to be quite quiet. There's not a lot going on, at least at this kind of large scale that we were looking at.
Yeah. And I can't resist to ask. So one of, my favorite types of mutations are the transposable elements. If they jump around the genome, interrupt stuff. But it seems like this is probably maybe not the right set of species or the right location to look for somatic changes. I guess maybe like if you had koala bears in there, which have active endogenous retroviruses, or if you were looking, as Vincent was pointing out, if you're looking at neurons, brain tissue, there's some provocative ideas about how somatic mutations are actually somehow involved, even potentially adaptively. And as neurons are sampling connections, some are killed, some like survive it through to sort of chisel out what the brain connections will look like. But yeah, I mean, I guess maybe this just reveals how, like, even a really big comprehensive study like this is just the tip of the iceberg. There's so many different directions to go. Yeah,
absolutely. So I think it's really just the beginning. And, we just say somatic mutation, but we really were limited in this study primarily to looking at single point kind of or small indels, insertions, deletions as well. But there's so much more to look at. And I think as well, tools like long read sequencing will really open this up in the future. So we were doing short Illumina reads, but when you get these like megabase or kilobase long reads, you just have so much more confidence in identifying and calling these retrotransposon events or structural variants. And I think redoing these kind of studies in the future with accurate long read technology will just have so much more to say about those kind of questions of larger structural variation.
Yeah, no, really cool. But a lot you can do with the point mutation. So, for example, you start to look then for signatures of positive selection, other things. So, what stood out in the data to you?
Yeah, so in terms of positive selection, not too much to say because the results are very consistent with kind of neutral evolution, which is in itself is interesting, but it's consistent with what we saw in human crypts. So it seems that most of these mutations that are happening are not having an effect, we think, on cell fitness, mostly neutral. And I think a really interesting thing about somatic mutations seems to be that cells seem to be very tolerant of mutations that you might think there'd be a lot of deleterious mutations that once they happen, the cell just can't survive. But it seems these cells can accumulate a lot of mutations without really caring too much. They seem to be fine. Although a caveat to that is I think we still have a long way to go in actually phenotyping these cells. So, histologically, these cells all look the same, but are they really the same? Are there differences there? And, that gets into understanding their effect on aging and, how can the cellular level, how can we really assess variation that's occurring?
Yeah. Yep. So, and then you draw some really cool comparisons. So I think and actually we've hinted at this, so getting back to Peto's paradox, et cetera, you point out the comparison between the giraffe and the naked mole rat. So similar lifespans, which is pretty wild for a tiny animal like a mole rat. But also similar mutation rates despite these huge differences in body mass. So what was that a big surprise to you? Actually, were you thinking of going the other way?
Yeah, it's easy in hindsight because the project's been going for years to think at the end, this all makes sense. But actually, I think at the beginning, I was really expecting to see more of an effect on body size with this whole Peto's paradox question. So then I would have expected that a species like a giraffe would have an even lower mutation rate than a naked mole rat, because although they have the same lifespan, you've also got to compensate for the fact that the giraffe has so many more cells. But we don't see that. I mean, the main thing that we see, the kind of take home from the paper, is that lifespan is a very strong predictor of somatic mutation rate, such that for these 16 different species, even though they have very different lifespans, they're ending their lifespan with a very similar number of mutations in their colonic crypts. So that kind of mutational clock is ticking at a faster rate in shorter lived species.
Yeah. Do you think that's what's going on there? Is there better DNA repair in the long lived species, or what's happening?
That is a great question. So and we can't really answer that from the paper itself, but we would speculate, that there maybe are more effective DNA repair mechanisms. And there's some fascinating papers from other groups looking at, I think, adaptive evolution in the SIRT6 protein, some of the proteins involved in DNA repair that seem to have accumulated kind of adaptive missense mutations in the longer lived species. So I think that's certainly kind of the next step one of the next steps of this research is understanding, okay, these longer lived species have lower mutation rates.
Yeah.
How has that evolved? What are the mechanisms? It seems more tempting to me that rather than these longer lived species having less damage initially, because all these species share the basic biology of the mammalian cell, the longer lived species have just found more effective ways.
It would be interesting to get polymerases from each one and measure actual mutation rates, right, as part of it. Because, as you said, there are other post-replication events that cause mutations, but that would give you, I bet they'd be all similar, right, if you did that.
Probably. It'd be fascinating to see, actually, a nice characterization across.
What about mutation rate versus genome size? Is there any relationship?
So that's something we really wanted to test, but unfortunately these mammals seem to have very similar sized genomes. There wasn't enough variation for us to really assess that. There's kind of two questions that we're really interested in linked to that. One is genome size, and one is, well, we see these similar mutation rates across the species. Is that because of is what matters just how many mutations you have, or does it matter how many kind of functional mutations you have? And then how do you define the functional genome? So kind of one proxy from that, from somatic evolution, we see in cancers is that most driver mutations tend to be in coding regions. So we could try using coding genome size as a kind of imperfect proxy for functional genome size. Again, unfortunately, mammals have similar coding genome to whole genome size, but that's what we're planning for kind of future studies, is to look at species with very different proportions of coding to non-coding DNA, and then try and see, do these correlations hold, and does it matter what actually the rate is in the coding genome? And that would really tell us a lot about the underlying mechanisms that kind of evolution is shaping here.
Yeah, because for viruses, the smaller, the bigger the genome, the higher the mutation rate. And
interesting.
Then it is a line. If you're looking at mutation rate on a y-axis and genome size, going from big to small, the line goes down. Mutation rate smaller with smaller genomes.
Yeah, and that's interesting to think about. I mean, the viruses are generally there, kind of getting to your point, Alex. I mean, they're pretty gene rich throughout, even with the ones with the big genomes. And so that could be a different scenario. I guess I'm thinking about, like, what's a massive genome? The axolotl, maybe the, Mexican salamander is sort of one of the, but there, as you're pointing out, it's like tons of repetitive sequence. And so what you really want to sort of maximize is that, like, maybe the sizes are the same, but the amount of coding space, the proportion of coding space versus non-coding space, which is maybe the easier analysis to do. Yeah. That's cool. So I guess the implication of the result then, this hypothesis that, if the mutation rates are keeping down in the long lifespan, is that there could be a link between somatic mutations and aging. So maybe walk us through that a little bit, or your thoughts on that possibility.
Yeah, so I guess that's the kind of the main take home, is that these results are consistent with somatic mutations playing a role in aging. And then it kind of gets the interesting question of, well, okay, that's interesting, but how? Like, how would somatic mutation rates play a role in aging? And we certainly think, the evidence supports that aging is a multifactorial process. There's telomere shortening, loss of proteostasis, epigenetic changes. So kind of the interesting, and the way we frame the question is, well, what particular parts of the aging process are somatic mutation contributing to, and through what mechanisms? So I think there's a lot of work to be done in figuring out, are all of these processes contributing to the same phenotypes in small amounts? So do you have all like epigenetic changes, somatic mutation all contribute to wrinkling of skin, or are certain phenotypes of aging each one kind of more or less due to certain processes. And so there's a long road to figuring that out. But kind of this kind of two main hypotheses we think about with the somatic mutation and aging. So the original hypothesis for somatic theory of aging is just that each cell is accumulating mutations. And as they accumulate mutations, it's decreasing their function and their cellular fitness. And eventually all of your cells across the body have just kind of taken so many kicks and knocks that they just stop functioning, and you kind of have this aging process. But there's some kind of studies that shed doubt on that hypothesis. There's been some recent studies of hypermutators. So you have humans who've got knockouts in polymerases that mean they accumulate a lot of mutations. And there's caveats. There's not many of those individuals who've been studied. So it's we can't confidently say that they don't show accelerated aging, but they don't always seem to, which kind of raises doubt on the idea that it's just having a lot of mutations that causes aging. But there's kind of another way of looking at it that we're interested in based on some of the work in humans, which is this idea of clonal expansions within cells, within tissues. So it's not necessarily that every single one of your cells has to just accumulate mutations, but if you are accumulating somatic mutations as you age, some of your cells may pick up mutations that actually change the phenotype of that cell, might cause them to expand more rapidly and colonize a tissue, but at the same time decrease their ability to carry out the task they're meant to do in that tissue type. So, and we see this in different cell types in the body from recent studies, that you do get these clonal expansions that look a bit like, that they're not cancers, but it's the same way of cells expanding, and they have knockouts in mutations in important genes. So we think that maybe as you age, the tissues are just losing their function because there are these spreading colonies of cells that aren't doing their job properly. And so it's not necessarily that every single cell is just accumulating deleterious mutations, but you have these evolutionary dynamics of spreading populations of cells. And this clonal expansion model could then kind of link into the observation we see of this, why evolution is limiting mutation. It's to stop these kind of deleterious clonal expansions as you age, so that you can reach reproductive age and do everything that evolution wants you to before the tissues start to fall apart.
Wow. That's a really interesting idea. I guess it could even maybe a knock-on effect of these clonal expansions is it could also sort of tie up the immune system or perk up the immune system. So you're not at a cancerous state, but you're still, there's some sort of an inflammatory process to so that this doesn't cross over to cancer in some cases, or the immune system's a little confused somehow, that why are there all of these things here that weren't here 20 years ago or whatever. I think that's a really interesting idea.
Yeah, so exactly. There's these kind of clonal expansions could contribute to de-differentiation of specific cell types. The tissues are these beautiful architectures of different cell types doing different jobs. And if you just get a expansion of one cell type, that's going to disrupt that balance in the tissues. So but absolutely, the role with the immune system is something that there's clearly interesting interactions between the immune response and somatic mutation in cells that we'd love to explore further in the future.
Yeah. And then, you mentioned, so you had to take great care to distinguish between the germline mutations, which are basically subtracting out from your analysis in order to get at the somatic ones. But then at the end in the discussion of your paper, you kind of draw a link between the process of germline mutation and somatic mutation. Maybe walk us through that a little bit. It's a really cool evolutionary implication there.
Yeah, so we see in humans that there are different mutation rates in different cell types, and we see that one of the cell types with the lowest mutation rate is the germline. So we did a cool study in humans where we looked at mutation rates in spermatogonial stem cells in the testes, and they are only getting, in the colon we're getting like 80 mutations a year, but in the spermatogonial stem cells it's only 2 or 3 mutations a year. It seems like a really protected cell line, which makes sense because you really want to protect the germline. That's what's being transported. But so then when we look at these somatic mutation results, one potential explanation could be that it's really the germline that matters. And there's a theory called disposable soma theory, because in a sense, the soma is really just disposable. You just need it for your lifetime, then it's gone. But the organism really has to care about protecting the germline. And so it's possible that selection is operating to lower the germline mutation rate, and then the mutation rates that we see that are higher in other somatic tissues are just a consequence of that. So whatever level of mutation rate that's selected for in the germline is what matters. And then obviously in a longer lived species, the germline will be more protected. There'll be a lower mutation rate. And that's why we could potentially see lower mutation rates in somatic tissue too, because it's just kind of correlated with the germline rate. So that's possible, that it's the germline rate that matters, and the correlation between somatic mutation rate and lifespan is a byproduct of that. But there's a couple of reasons we don't think that's the case. One is that, the key one really is that the mutational processes we see operating in the germline aren't exactly the same as in somatic tissues. So the ratio of those mutational processes operating, and there are certain somatic processes we only see in somatic tissues. So if the mutation rate was it was only the germline that mattered, we'd probably expect to see, well, if you saw the exact same processes across all somatic cell types, there might be a bit more likely that that's the case. But we actually see that it looks like evolution's kind of fine-tuned different mutational processes in different somatic cell types already. So why there would have to be this tight correlation between mutation rate between the germline and the soma is kind of, and it would be a question you'd have to answer.
Yeah.
So it's certainly possible, but yeah, I think exploring this relationship between germline and somatic mutation rate will be fascinating. And there's interesting species where there's not a clear distinction between germline and soma that would be interesting to look at. I mean, so I think again, that's why I'm so keen about looking at other species, because you can start to tease apart these questions.
Yeah, that's great. And also, I think generating a lot of interest in the field exactly along these lines. So having this first comprehensive data set across 16 mammals to start to sketch out this sort of opens up the possibilities of comparative stuff in the future. Really cool. So we've talked about, I think, kind of a lot of, possibilities for what's next along the way. And you've mentioned this and highlighted that too. How would you prioritize? There's so much to do, Alex. How would you prioritize the work ahead?
Yeah, there's definitely a lot to do. So I think, I think what we're interested in initially is extending this to species with an even greater range of lifespan and body size. I mean, we see this correlation with lifespan in mammals. Does that hold further across the tree of life? That's kind of a main question. And that's appealing because we can kind of, keep applying the methods we've already developed, just extend that. So looking at different vertebrate species, particularly interested, in fish, where you have some species with a lifespan of a couple of months versus sharks that are potentially living hundreds of years, like the Greenland shark. So kind of trying to draw these correlations with lifespan across different groups in the tree of life.
You could do that in plants too, right?
Exactly. And there are some nice studies looking already, like an oak tree somatic mutation rate. The challenge is always, it's a shame, well, it's obviously not every species has a clonic crypt, so we'll have to start looking at carefully selecting different tissue types. Another limitation of the study was that we only looked at one cell type. So within mammals, we want to go back and, as the technology is improved and we have a better understanding of tissue architectures, look for other cell types that we could kind of apply the same approach to. So potentially blood, liver, and just see, do we see the same correlation with lifespan across tissues? Or is it something that was just happened to be unique to the colon? So that's kind of one direction.
Yeah, really cool. I think that covers the paper pretty well. Vincent, did you have other?
Pull in some listener questions here.
Yeah, good idea. Yeah.
Andrew wants to know, are stem cells more significant in cancer risk than normal cells with regard to somatic mutations?
Well, that's a good question. So I think there's a lot of controversy within kind of the somatic field about what constitutes a stem cell. So I think there's a lot we still don't know about tissue types, about the exact architecture and what is a stem cell versus a differentiating cell. So I think it would depend actually cancer type to cancer type. So the different tissues have very different mutational profiles and different risks. So I don't think there's a clear yes or no answer to that one, but it's kind of, as we learn from different cell types, where the cancer is coming from. And I think there's still work in understanding what is the cell type of origin for some types of cancer. So, and there's different hypotheses. So I know some work, for example, in pancreatic cancer, where we're trying to understand in ductal pancreatic cancer, is it actually the ductal cells where the cancer comes from, or is it that you have other cell types that then differentiate and become ductal, and trying to trace back that phylogeny to understand what are the cells of origin in cancer. So there's still lots of exciting work going on in those kind of questions.
I mean, the other issue is your colonic crypt cells are dividing constantly, right? They're one of the few cell types in the body, and it would be interesting to compare that to some that don't, like neurons or other muscle cells, right, that don't divide all that much.
Exactly. So that was a limitation at the beginning of this study, was that we chose colonic crypts because we needed to kind of have these dividing cells. But now we have new duplex sequencing methods that free us from that limitation of clonality. Great. And so, we'll be looking at neurons for sure.
So Rune wants to know, if salamanders have somatic mutation, they're able to regenerate limbs, etc.
Yeah, that's. I mean, I think every organism has somatic mutation. But I've been thinking it would be fascinating to look at some of these like axolotls where they have regeneration and see kind of what's happening at that frontier of regrowth, because the kind of clonal expansions that we see driven by mutation in healthy tissue are probably also happening at these regrowth. Are they more prone to clonal expansion of kind of mutated cells? Fascinating to look at. Yeah,
that's a great question, Rune.
Yeah, it's that's it for our listeners for now.
Sweet. Yeah, great questions. So before we let Alex go, I really wanted to spend a few minutes talking about science illustration, science communication. So at evolution meetings, Alex kind of first came on my radar. I think many evolutionary biologists sort of, Alex is famous for his live sketches. So shows up at a talk at a meeting and he's got his iPad. I guess this came in handy, your skills for outlining. You're using the same thing to outline your colonic crypts to guide the laser, yeah. Sort of like these echoes of different skills. But in this case, it's sort of a really unique and cool way. I think maybe it's been copied a few times now, but a unique and cool way to record a talk. So walk us through that, Alex, how you got into live sketching and what you're doing there.
Yeah, sure. So there's something that kind of started during my PhD. So I remember actually there was a science journalist who was visiting our lab at some point during my PhD who mentioned this thing called Twitter that I'd never heard of, and mentioned, "Oh, you should get on Twitter. It's great." And I was going to a conference like 2 weeks later, an SMBE meeting, I think, and downloaded Twitter. And I was at talks with my little iPad Mini, and I realized, people were live tweeting talks, which is great, and people kind of tweet along, and you can see, especially in these conferences that are huge, you can't attend every talk, so you can follow what's happening. And I wanted to contribute to this somehow. And I realized I like to doodle when I take notes. And I realized that actually tweeting a picture is kind of a cheat to get around the Twitter word limit. So if you tweet an image, you can put as much text as you want. So it really kind of started that way. And then as it evolved, they were really just doodles, title of the talk. But I did try and include a little sketch of the speaker, like a very quick doodle, which I think I've always found is a part of science communication I like to include is the scientist as well as the science to kind of help people engage with it. I find that if you just have the science without the speaker, sometimes it's a bit harder, especially people who aren't there, to be drawn in and engaged to the kind of the human side of it. So I always tried to draw the speaker and kind of the highlights of the work. And over time, that's just kind of evolved to be a bit more artistically interesting, not just a doodle, but try and have something that's like nice visually to look at. And yeah, it's been great. I used to just do it when I would go to conferences that I was going to anyway, and as a way to record it. But because it kind of got popular, I ended up getting invited to Illustrate conferences, which has been really fun. And I've actually, I didn't mention this in how I got started in my postdoc, but one of the things that I remember now that got me interested is I was invited to a conference on somatic evolution to illustrate it. And while I was illustrating it, I realized how cool this was. And so that actually is a way the illustrations influenced my scientific career as well, just by giving me this opportunity to be exposed to different fields of science that I otherwise wouldn't have been so closely connected to.
Yeah, let me show really a couple of examples. I found, I guess this is your website, Alex, with...
yeah, that's right.
This is the idea, right? This would be a single illustration and with your notes and pictures of the people involved, right?
Exactly. And I do them live just partly for time constraints. If I didn't do them live, I just wouldn't have enough time to get them all done. And it also, I'm very prone to my mind wandering, so it's a great way to actually keep me focused on a talk.
Yeah, here we have some of the animals we talked about today: lemurs and whales and elephants.
Exactly, yeah.
Oh,
that's a group that looks at cancer rates.
It's great, very cool. Yeah. So, how the mechanics? You're doodling on your iPad, and then you save it as a JPEG, and then post it on Twitter. Is that how it goes?
Exactly. Yeah, I just live tweet them out as I go along usually.
Wow.
Precisely.
Well, helps to be an artist. You're good. I could never do that.
But if you scroll back to my very beginning, there's definitely you see a journey of increased quality over time. So I think it's just having the opportunity to practice. So I would encourage anyone who is interested to just get going and give it a go, because you don't have to produce wonderful illustrations for it to be useful and interesting. And even I find it useful for myself, even if I'm not sharing those images. Taking notes and adding visual elements for me works really well, just for remembering the content of a talk. So, and I think different people have different ways that work for them in note taking, and it would be great, I think, on Twitter to see more people's take on their note taking in these kind of ways, and just to help. It's good for people who do it, and it's also great for people who can't attend talks to see.
Yeah.
I just put the link in the chat so that people can have a look how cool these are. It's just great.
Yeah. Really cool. And I think there is, I don't know how to define this or if it is even really a thing, but something about, when we're drawing or doodling and how it sort of opens up, it unlocks a part of the brain almost that I think is perfect for when you're thinking about scientific ideas in new ways. It's sort of that almost daydreaming state somehow coming together. I think that works for me. So I'm not nearly as public or prolific of a doodler as you are, Alex, but if I go through my lab notebooks actually, or if I'm listening to a talk or on a meeting on Zoom, if it's like an hour or something, I'll come back and there's like pages just filled with like ridiculously weird scribbles and doodles that are not nearly as interesting and professional, but it's somehow there's an important part of the science process. It helps unlock something.
Yeah, I think absolutely. So also when I'm not drawing it for an audience, my notebooks are just full of just random doodles that aren't always even connected to the topic. But and I used to definitely think that was a hindrance, that I'm being distracted. But actually, I do fully embrace now that actually just letting your mind and your hand or whatever doodle as you're listening, actually it does do something that's positive, and I've embraced it now, that it's just kind of giving space for your mind to think somehow and draw connections. And I think I don't think anyone should be ashamed of doodling. I think it's actually helped to think.
Agreed.
What app are you using on the iPad?
Yeah, so for the live illustration, I use an app. It's called Paper, so it's easy to remember. By Paper 53, but the app's called Paper. There's other better apps out there for drawing, but this is the best one for drawing quickly. And when you're doing a 10-minute talk, speed is of the essence.
I think that should be your pick for today.
Science pick of the week. Yeah, we'll put that up for sure.
Paper.
Before we get to our picks, I can't help but ask one more question, which is, your artistic skills have also are showing up not just on your live sketches and on Twitter, but also on the cover of Nature this week. So maybe actually, I don't know if you can grab that, Vincent, if you can go to the link there, and throw that. Maybe walk us through a little behind the scenes here of how you ended up contributing an illustration that is actually on the cover of the magazine.
Yeah, sure. That was a nice research kind of career highlight for me. So that was a great process. So when you get an article accepted in several journals, they'll write to you and say, if you have an idea for the cover, send it in and it'll be considered. And it's always a bit, it's a bit of a competition because several authors will be sending them in, and you never know if you're going to get it on the and this is the success example I've had many times in the past where the cover idea hasn't been accepted. So this is nice to finally have it. So they get back to you at the journal. They make I sent a few different potential designs in. There was one they liked the most and they adapted it a little bit, and then I kind of worked back and forth with them. I'm really happy with the final result. Wanted to get across an illustration that kind of summarized the key concept of the results, but also was visually appealing to look at. And I think definitely the illustrations do help to communicate the science. And I would say, even in the past where the cover hasn't been accepted, you can still use the kind of cover illustration you come up with to kind of share the article on Twitter. And it just helps people to, it's a busy social media marketplace, and getting people's attention to see the research.
Yeah, I agree completely. And you're right. Like this notion of like the competition for the covers. I mean, so I've got among my folks in my lab who we've made many cover ideas over the years, none of which have seen the cover. We've actually been thinking about launching a journal of failed cover images. And
that's great.
So
that's going to be awesome.
Yeah. And I'm putting it out there to the world because I've been thinking about this for years and haven't delivered on it. But so basically, I mean, the trick is, who do you put on the cover of the journal of failed cover images?
Covers,
but every page is a different cover idea. And then and then a paragraph just saying, what are you trying to capture here? What's the science? And then a link to the actual paper as a way of sort of, trying to connect a little bit of the artistic side and to the scientific side. I think that's like really fun. But congrats on this success. It's a really cool image that deserves to not be in the journal of failed, but actually in the journal. That's really cool.
Thank you.
Here's a good comment. Try capturing the magnificence of Nels's beard in a doodle. Impossible.
Yeah, that's some things you can't can't illustrate. You just have to let go. That I would submit that's one, but that's pretty funny. Well, Alex, this has been great. So I think why don't we move on to our picks of the week? So and we'll ceremoniously add the paper app as Alex's pick. Sure. This is the application that you've been using for your live sketches so effectively over the years. So my science pick of the week is the 50th. So there's a special issue of Philosophical Transactions of the Royal Society, B, and it's the 50th anniversary of Dick Lewontin's this paper that was got a lot of attention, the apportionment of human diversity. And so we've been talking about somatic mutations. We've been talking about mutations. This is getting at sort of more of the germline or the population diversity. And one of the really important, parts of this paper, the reason that it's being commemorated and celebrated, is that it points out that 85% of human genetic variation resides within populations. And this is important to remember because it really dispels the notion of sort of of a biological definition of race. This is, as we've been talking about recently on TWiEVO, race is true, exists as a social construct, and not a biological one. And so that's important because some of the arguments that are being sort of falsely hoisted based on that misconception have moved things in some not great directions. So it's a really cool issue, and I'll have the link up here for my pick of the week that then has a number of review pieces, sort of like 50 years on, how are population geneticists thinking about human diversity, human variation. And it covers both from just continuing to move the science forward, kind of as using still in the echo of this landmark paper by Dick Lewontin, but then also all the way to some essays getting at what I was describing before, sort of, how do we think about human variation and put that into a good context. So that's my science pick of the week. Vincent, how about you? What's your science pick of the week?
I have a very brief blog post over at Small Things Considered by Roberto Coulter. It's called An Exceptional Vintage. And I like it because he talks about having a dream, but he first starts by saying it was influenced by a paper he read which describes the fungal populations of Corvina grapes, which are used to make Valpolicella wine. And these are grapes that are allowed to wither on the vine after they're mature. And that, of course, concentrates the sugars and so forth, and they become very full-bodied wines. And more to the point scientifically, in this paper, the investigators found a core fungal community that assembles as they age on the vine, and this gives it the remarkable quality of the wine. So I like that part a lot. And then he goes on to describe his dream was looking for a bottle of a 1928 Amarone in Milan at one point, and this is in his dream. So I'll leave it to you to read the rest of them. But it's a lot of fun, and I like the juxtaposition of science and and dreaming.
Yeah, that's great.
Which is very cool. We also have a listener pick from Martha. Let me scroll down here. Where is this? This is at nope, that's the wrong podcast. Sorry.
Oh, no problem.
Here's Martha. Dear TWiEVOs, I enjoyed the live stream on the stink bug version of It Isn't Easy Being Green. That was our last episode. In that episode, you mentioned an interest in the history of infectious disease. Let me recommend Plagues Upon the Earth, Disease in the Course of Human History by Kyle Harper. He's a historian, but starts in prehistory, with prehumans working his way to the present as our species acquired different infectious agents. I was reassured early on that he had done his research when he quoted Dr. Racaniello. Also available as an audiobook. Best wishes, Martha. Thank you, Martha. That's cool. I'll have to check that out.
Agreed. Yeah, that was such a fun paper we did last time. This was basically the authors replaced a bacterial symbiont in stink bugs with E. coli, and it took one mutational step for the stink bugs to sort of regain some of their important sort of essential biology, including this distinct green color. That was really fun.
Well, that's cool.
Pretty wild results there. Yeah. Well, speaking of fun, this has been really great, Alex. Thank you so much for joining us on short notice, for walking us through your paper, for sharing some of your illustrations with us across the board. Just really great. Can't wait to see where the research goes. Can't wait to see where your career goes. Keep up the great work. So exciting.
Well, thanks so much. It's been my pleasure. It's been really fun to, share the results and talk about it a little bit. So I hope to come on again in the future, maybe with new results one day.
Anytime.
Please do. Yeah.
Anytime. We love it.
Just reach out.
All right. You can, this is TWiEVO 77. You can find the show notes at microbe.tv/TWiEVO if you want to send us a question or a comment. TWiEVO at microbe.tv, or just show up on the live stream and ask it there. If you really like what we do, consider supporting us. Go over to microbe.tv/contribute. Your contributions, at least in the U.S., are federal tax deductible because we're a nonprofit corporation. We depend on you to be able to do this. Our guest today from the Wellcome Sanger Institute in the UK, Alex Cagan. Thanks so much, Alex.
My pleasure.
Nels Eldies at selvolution.org, Early Bird on Twitter. Thanks, Nels. Good to see you again.
You too, Vincent. I apologize. I was kind of sort of going in and out here with some internet
worries.
Hiccups, but we did it. And thanks to the crew from all around the world who showed up here asking great questions, weighing in, and that's just so much fun. Thank you all.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.